As the population grows and the global standard of living improves, the demand for aquatic protein or seafood is increasing. Aquaculture, the farming of aquatic organisms, has been responsible for the continuing impressive growth in the supply of fish for human consumption. Today, aquaculture provides close to 50% of the world's supply of seafood and expected to double by 2050 to supply the increasing global demand. However, aquaculture faces a range of sustainability challenges. These include environmental and social problems. This video contains the research on sustainability in the aquaculture system from the first International Conference on Sustainable Agriculture and Aquaculture for Well-Being and Food Security. It also highlights the possibility that filling certain knowledge gaps could help to further improve aquaculture sustainability.
Good afternoon, everyone. I think Thailand is very good, nice weather now. Now I'm from Japan. We have a 2-meter heavy snow. So good afternoon, everyone. Again, we are really appreciate to have a great opportunity for having this presentation. I am Watari from Nagaoka University of Technology. Today, I'd like to talk about topic of development of DHS USB reactors for long-truth grouper closed recirculating aquaculture systems. This research is collaborate research with Kanazawa University, Japan, Chulalongkorn University, Thailand, and also National Science and Technology Agency, NASDA, Thailand. So let me introduce introduction of these studies. I think everybody know the closed recirculation aquaculture system, but it's a little bit introduced. This system is an insensitive fish farming technology used in the water restrict situation and can produce safe, healthy, and high-quality aquatic products. Up to 90 to 99% of water is recirculated in RAS with high efficient water treatment process. And this innovative fish farming technology approaching is increasing by applied for the solving food problem as demonstrated by several previous studies worldwide. This array system is in Japan. We also really interested to develop these technologies. However, we have several remaining issues. Strict water quality controls in order to ensure the survival of fishes at high density conditions. And deterioration of water quality in RAS can lead to the death of all cultured fish. Therefore, high-performance water purification system is required. So in order to solve this problem, we are using the downflow hanging sponge reactor that we call DHS reactor was used for the RAS water purification system. The DHS reactor was developed as a low-cost treatment system for developing countries, and we already installed a full-scale DHS system in India, Egypt. This reactor is used sponge as a biomass retained carrier, and it has high oxygen transfer without aeration. In the middle figure shows the schematic diagram of these systems. As you can see, the water is come from top of the reactors, then naturally fall down to the bottom. During that time, oxygen can naturally dissolve into the waters. Therefore, we don't need any mechanical aerations like an activated sludge process. Moreover, this DHS reactor can retain high concentration of biomass, exactly more than 10 to 20 times higher biomass concentration can retain in the sponge compared with conventional activated sludge process. And also, this sponge, inside this sponge, there are many bacteria or archaea or some microorganisms living there and have high biodiversity. And this high biodiversity can perform the low excess sludge production. So this slide showed our sewage treatment technology project in Thailand. Since 2018, we construct 3.6 cubic meters pilot-scale DHS system in Khon Kaen, Thailand, and evaluated its process performance until now. As a result, the DHS system can treat the low sewage concentration only 30 minutes, and the final effluent can achieve the discharge standard. Moreover, excess sludge production was much lower than conventional systems such as conventional activated sludge process. Now, we are planning to install the full-scale DHS system in the North Thailand. So, in this study, we applied this DHS technology combined with anoxic denitrification active upflow sludge bracket reactor USB for wastewater treatment system of RAS in Japan. To achieve this object, we evaluate process performance of DHS USB system for long tooth grouper RAS and evaluate growth of long grouper under the high density and different salinities. Salinity is one of the most important environmental factors affecting water quality and fish growth. These are reports that the biological treatment process under the high salinity conditions significantly affect to the system performance. Also, the total nitrogen removal decreased with the increased salinity due to the ammonia oxidation was inhibited. High salinity condition can interfere the ammonia oxidation rate and reduce cellular enzymes activity of microorganisms lead to the poor bacteria performance. In addition, affecting microorganisms which decrease efficiency of nitrogen removals. Salinities can interfere the mechanism that maintain the ionic and osmotic homeostasis of fish body fluids, which is affecting the fish growth and survival. So now I would like to explain the methodology of these studies. A pilot-scale DHS USS system consists of two DHS reactors and two USB reactors were installed in my universities. The USB DHS reactor was installed in the RAS system to maintain the water quality aquaculture tank. The condition in the aquaculture tank was controlled to provide a healthy environment for fish growth during the entire experimental payload. The fish used in this study is a long-tailed grouper. I cannot pronounce this English name, but we call it kue. And initial number was 305. The water from the aquaculture tank was pumped directly to the top of the DHS reactors. The effluent from the first DHS reactors was disinfected to the UV lamp, then recirculated into the aquaculture tank. The USB reactor is a denitrification reactors. Acetate or sodium acetate was fed as a carbon source. In this study, the system performance and the growth of the fishes were evaluated for the over 800 days. The operational condition of the DHS USB system consists of 3 different salinity payloads. Salinity was gradually reduced to 30 Bermil, 22.5 Bermil, and 50 Bermil during the study. The retention time of each reactor was shown in the table. Next, we would like to move to the result and discussion. This slide shows the water quality in the aquaculture tank. The performance of the aquaculture tank were evaluated based on the total ammonia nitrogen, nitrite, and nitrate concentration. From the result indicated that DHS reactors has available to maintain the total ammonia nitrogen concentration. The less than suggested value 1 milligram nitrogen per liter at the various salinities. Nitrate is not toxic compared with the ammonia and nitrate. However, high nitrate accumulation in the RAS can lead to the inhibition of fish growth and further long-tooth grouper can survive a maximum nitrate concentration of approximately 500 milligram nitrogen per liter. This slide shows the performance of total ammonia nitrogen nitrate in the USB DHS system. From the result, show that high nitrate and nitrite concentration of salinity of 22.4 barmy and 15 barmy. The nitrate and nitrite concentration were increased from the USB effluent because of a pump problem, and so solid state feed was not effectively. However, the results show that the DHS system can able to maintain the total ammonia nitrogen concentration at low concentration during the entire experimental payload. Next slide shows the growth of the long-tooth groupers. The length and weight of fish during the change of salinity at 30 barmil and 20.5 barmil and 15 barmil shown in this slide. All the fish grow and survive during the experimental payload with an average daily weight gain of 0.8 salinity, 0.8 at salinity of 30 barmil, 1.3 at salinity of 22.5 barmil, and 1.9 gram per day at salinity 15 barmil. From this result, indicated highest growth rate was obtained at salinity 15 barmil. So this is the photo. The up photo is a initial long-tooth grouper, and we conducted high density and high nitrate experiment. The growth rate is equivalent with sea surface aquaculture, and finally, it now getting nearly 50 centimeters long-tooth groupers. And also, we ate this long-tooth grouper is like sashimi or hot pot. It is very delicious. And now we are planning to sell this fish to the restaurant. So this slide shows the conclusion of this study. The DHS USAV system has able to maintain total ammonia nitrogen concentration in the aquaculture tank less than 1.0 milligram nitrogen per liter. Salinity in the range of 15 to 30 barmil didn't affect the performance of DHS USAV systems. And also, we observed the highest fish growth rate was obtained at salinity 50 barmil. And now we have a still remaining issue for this research. The first is upscale. Now we are do this experiment at 5 cubic meter tank, but in order to make industries, we have to increase more upsize. And at this moment, we are the system was operated by the 2 or 3 students, but for the business, maybe we have to do the more mechanically to do the easy operation. And also, in my university is located in the north of the Japan, and always we are cold. So therefore, we use a lot of electricity for increase the temperature in the tank. Therefore, we have to consider the reduce the thermal cost. So this is my presentation. So if you're interested to my research, please contact me. Thank you so much.
I would like to know, have you checked the group of the bacteria in the sponge after proper culture.
Have you checked the microbial community?
Thank you so much for your question. At this conference, I didn't show the microbial community analysis, but I already analyzed the communities. In the different salinity conditions, also the microorganism in the reactor is changed. This is shown in the family level, and this is and the most interesting, important things is nitrophene bacteria community is little bit different. When operated with middle salinities, we detected a large amount of nitrosphere. That is very important because this is nitrite oxidizing bacteria. And in case of the 30 bar-mil, the nitrophene bacteria detection rate is very low. So that's why we are recommended to the operator with more low salinity condition to the for the like a good for the bacteria. Yes.
Hello, good afternoon, everyone. I am Evelyn, and similar to Dr. Watari, I am from Nagoya University of Technology. So today, I would like to share with you about my study, which is the development of a prospective zero water exchange system for aquaria using the Ozone DHS USB system. So first, let me tell you a little bit about the background of my study. My study deals about developing a less maintenance system for aquarium. But do we need that? Oh yes, we do, because an aquarium requires regular maintenance in order to maintain the water quality and its aesthetic beauty. What makes the aquarium, but actually what makes the aquarium dirty is the fish itself. Fish releases nitrogenous waste through respiration, excrement, and also via uneaten food that later become ammonia. Ammonia is highly toxic and harmful to the aquatic animal. So if no action is taken, it can lead to fatality. Currently, the most common conventional methods used to address this matter are using water exchange or filter, especially sand filters. However, this current maintenance method has few drawbacks, such as for water exchange, it requires frequent work, at least one or twice a month is needed to cleaning of aquarium is needed to maintain the water quality and also its aesthetic beauty. Next, it also uses abundant of water. It consumes time and also demand a lot of effort. For sand filter, especially, it also requires regular work because over time it can be clogged and it needs backwashing. And again, due to backwashing, we will use abundant of water. And then in addition to that, it also takes space. Sand filters are usually bulky, so we requires like quite a spacious place when we use sand filter. To overcome these issues, we develop a prospective zero water exchange system for aquaria using the Ozone DHS USB system. Our objectives are, first one is we want to study the suitability and efficiency of ozone DHS USB system in removing color and nitrogenous compounds such as ammonia, nitrites, nitrates at long-term operation. The second one is to investigate the impact of moderate ozone concentration on the performance of the biological DHS USB system, because ozone can also be toxic not only to the microorganisms in the reactors, but also to the fishes, living fishes especially. So it is also important to investigate whether this system is suitable or not. The study was conducted using a 700-liter aquarium bred with 3 ornamental carps, Nishiki-goi, at the top tank. The bottom tank consisted of a DHS system, which is responsible for nitrification, and USB system, which is responsible for the nitrification system. Generally, the water in the top tank, which contains ammonia, released from the fish, is designed to overflow into the DHS system. Here, the ammonia is converted to nitrites and nitrates by the microorganisms in the DHS reactor. And then it will be released into the bottom tank. The water in the bottom tank will then pump up into the USB system and be converted by the microorganism in the USB system, a USB reactor, into nitrogen gas. Because nitrogen gas is inert, it can be released into the air. The water can be recirculated back into the top tank. But before it is like recirculated back into the top tank, the water flow through an ozone section to remove color of the water. Among the advantages of this system is it can be made in compact size. It doesn't require aeration, so we can reduce electricity cost. It reduces water exchange, and it requires less maintenance. So these are the DHS and USB system at the bottom tank. The study was performed in 5 phases for 600 days. The first 2 phases, start-up and phase 1, only consisted of DHS USB system. And then from phase 2 onward, ozone was applied together with the other 2 reactors. But only phase 2 and 4 were operated with the presence of ozone. We also perform microbial community analysis using the next generation sequencer, Illumina MySeq, for further understanding about the bioreactors. Next are the results. So the results will go according to this. The attainment level for this study are ammonia 0.1, nitrite 0.1, and nitrate 10 milligram nitrogen per liter. The ozone DHS USB system has successfully maintained all 3 nitrogenous components within the attainment level. Based on the card body weight, a potential up to 1.5 gram nitrogen millicubic per day of ammonia was released into the water. Supposedly, the concentration of ammonia increased in the water, as you can see, the black line at figure 5b. But with this system, the average ammonia concentration in the aquaria was successfully retained below the target level at 0.1 plus minus 0.2 milligram nitrogen per liter. The results showed that the ozone DHS USB system has successfully maintained the water quality in the aquaria at long-term operation. Most importantly, we eliminated the needs of water exchange. Water exchange was performed only 4 times, 1, 2, 3, 4, during phase 1, and it is to dilute the color of water in the aquarium. Accumulation of yellow substances due to limited water exchange tainted the color of the water. So the water needs to be exchanged because aquarium requires clear water for display. After ozone was introduced, water exchange was not performed anymore for 425 days. Next is the color removal. Color doesn't harm the fish. However, an aquarium with a purpose for display required clear water to maintain its aesthetic beauty. It is observed that the color of water in the aquarium reduced about maximum 550% during the presence of ozone. The water looked much more clear by eyes and therefore eliminated the needs for water exchange. For further understanding about the reduction of color in the aquarium water samples were collected from the aquarium, and the water were aerated with air and ozone. These experiments aimed to identify the components existing in the water samples and understand the ozone mechanism in reducing yellow substances in the water. Based on the emission excitation wavelength, component 1 seemed to be humic-like substances, and component 2 seemed to be like protein-like substances. The results showed that no obvious difference were observed at both component 1 and 2. Humic substances are habitual components of the organic matter in the water. Unfortunately, its presence has been reported to affect the color of water. Therefore, the elimination of such substances is important to maintain clear water in aquarium. Next is water samples aerated with ozone. Reduction of components of humic-like substances were observed when in the ozone aerated samples. Component 3 and 5 suggested that ozonation aided in oxidizing humic substances, and thus the reduction in the presence of yellow substance over time could be explained. Next, we also analyze microbial community in the reactors. And here, ammonia oxidizing archaea and nitrate oxidizing bacteria is likely to play a major role in ammonia oxidation process. And in the USB reactor, Tara seems to dominate the reactor. Tara, is next, the fish health and survivability assessment. Ozone concentration detected in the aquarium is about 0.03 milligram ozone per liter, and it is reported that about 0.1 milligram ozone per liter is lethal to fish, but no abnormal behavior and health effect on carps were observed. And we could conclude that most probably because carp have higher tolerance toward ozone toxicity. Overall, ozone degrades color in the water, and DHS USB system successfully maintained the water quality below a attainment level for long-term operation. Secondly, the impact of moderate ozone concentration on the microorganism in the reactor is ambiguous. However, not to the extent where it affected the performance of the biological DHS USB system. And thirdly, the fish survive at 0.03 milligram ozone concentration detected in the water without water exchange performed for 425 days. So we would say that this system has high potential towards zero water exchange aquaria and is a promising technology to solve water exchange necessity issues, provide water security, and also to lead maintenance-free system for aquaria. Thank you for your kind attention. If you have any questions, please feel free to contact me at the written email. Thank you.
My question is, I see you don't have a mechanical filter for like solids. What happened to all the solids that you accumulate in your system?
I see. Yes. For mine is slightly lower density, and Dr. Watari is higher density. For mine research, yes, we don't have a mechanical filter, but the DHS reactor itself, it consists of sponge. So when the solid flows through the DHS reactor, it was like kind of trapped at the top part of the reactor. So it acts as both, it's also a filter for solid and also treating the ammonia. And
you didn't have any problem with like clogging the matrix of the sponge?
For total operational time, 600 days, no, we don't have any clogging problem. And I didn't even like wash the sponge at all.
It has a little bit of similarity with the Dr. Watari and Dr. Adeline presentation now, because we're talking about the recirculation system. And I'm going to talk mostly about the sustainability of this kind of system and how we get to that sustainability. So first of all, important to understand about aquaculture. So we know that aquaculture is growing very fast. What happens is, by the 1950s, if you look at the blue curve, is the production of aquaculture. And by 1950s, 1960s, we know that like aquaculture has been done for thousands of years, but in the 50s and 60s, that's when actually the real research started, and publications about aquaculture. And people start noticing things and following trends and then learning better about aquaculture. So that's when the real aquaculture started, and that's how we can have this big growth. And I remember in the 90s and in the 2000s, that people, some people would say, oh, soon aquaculture reach fisheries. Many people didn't believe that it would reach, but actually by 2012, we surpassed the supply of fish from fisheries to aquaculture. So aquaculture now is the main supplier of fish and seafood to the world. And what happened for this to happen? So first of all, as I said, all the research done in the 50s and 60s, this started a big push of aquaculture, learning all the technologies. So this gave the increase in production, which we see till nowadays, production always growing, with the technological developments. So we learn more about water quality, about nutrition of the animals, and all these things. A lot of training, a lot of seminars, a lot of conferences like this one, like a lot of information being disseminated and people learning more. So this is very important, the technological developments. Rising income worldwide, everywhere people are getting a little bit more money, so they can buy more fish, and also cheaper fish and cheaper seafood, if we look. Other very important thing, reduction in loss and waste. So transportation, much better. The processing factories, more feed mills. All these made the reduction of loss and waste of all the fish produced, okay, for food. Another thing is the health awareness from the consumer. So consumer is the one that dictate what is going to be produced and what's going to be sold in the market. If a lot of people is looking for fish will be sold. There'll be more production, so there will be demand for the fish. So it's the market demand. So the consumer is the one that dictates this, and this is very good. And this started also in the 80s, 90s, and it's been growing very rapidly. So I'm going to show a little bit about this research center called Aqualis in Thailand. So this research center is very close to Bangkok. It's in Samut Sakhon, which is about 1 hour from Bangkok. And is a research facility from Diana Aqua. So this is the facility. This is the blueprint of the facility, about 1,000 square meter. And I'm going to explain exactly what is what here. So if we look, let me just put a pen here. So here's the main entrance of the facility. Okay, this is a ramp, it's a concrete ramp. We can come with the truck here to deliver fish, or even bring water if you need like concentrated seawater or anything with a forklift. It can come in here. So what happens? This is the first entrance of the facility. Then we have, right by the entrance, there's 4 reservoirs of water. So we use 2 for fresh water, which is city water, so it has chlorinated and everything. So we have aeration to remove the chlorine. We use also sodium thiosulfate to remove the chlorine. We can do chlorinate more if we need. We can use other chemicals to sterilize the water. Another 2 tanks are used for concentrated seawater, so we can distribute throughout the system. Now, outside, this is outside the facility, okay, it's not indoors. Everything else is indoors. This is a nursery and a quarantine area also. So all the animals that we receive goes into this area outside the facility to make sure there's no contamination, cross-contamination with the facility. So it sits there, we look at the animals, we keep them for a few days to see if everything is okay, and then we start sorting to go into the systems. And the systems are inside. Here are the systems. We have, this is one recirculating system, this is another recirculating system, and here it's an aquarium system. Okay, all systems have 32 tanks. The tanks, the plastic tanks here and here are 500 liters, and the aquariums are 150 liters, and everything is independent. So there are 3 independent systems recirculated, and I'll explain a little bit more how they work. Next thing, outside the facility, all the water used inside the facility and filtered from the facility, from water exchange or anything, goes into this canal outside. This canal will have a aquaponic system, and I'll show more information later. And the last step for this water, so the whole system here is closed inside. All the excess, if there's any excess or wastewater, goes into this canal. And then we have plants to absorb the excess nutrients. From this area, it goes into this oxidation pond here, which is the last stop of this water. The water doesn't go into the city water, the sewage, okay? This is the last stop. It doesn't go anywhere else other than here. So this research system is used for in-house testing. So we use for palatability testing, for nutrition tests, to see different products, to develop new products, to benchmark products. Also, we use it for commercial trials. This is not open for just being contracted. It's like our customers, we do small trials with them. We can test their feed, tell them what's the performance of the feed, all these kind of things. And the last mission we have is species knowledge. So any new species, for example, if you're talking about climbing perch or even some different species of snakehead, and we want to learn more about the species, what kind of feed is good, what is palatable for them, so we can do that. And this is the next step also for the future. We're still working mostly on palatability of tilapia and shrimp that we know. We're going to start working on sea bass and other species that are more commercial, and then we're going to go into the species knowledge in the future to try to bring more information also to the market. So here's the entrance on the left side, the ramp that I said. You can come with the truck or with the forklift. This is the facility. It's all indoors. It was finished, the construction in 2019 in December. It's about 1,000 square meters. We can have fresh water and seawater inside. 3 aquaculture systems, recirculated. This picture on the right side is one of our technicians. This is the outside, the nursery, the quarantine area. He's collecting the tilapia, the red tilapia, to go into the systems to do the trial. And this is inside the facility. So here we see 2 of the recirculating systems, 32 tanks on each one of them. And they're independent, as I said, so 500 liters. Those are big plastic tanks. Okay, so we use, for this system, we can do 8 treatments with 4 replicates. We make sure we always have 4 replicates. In case we lose 1 replicate, we can still do statistical analysis. So we make sure to always have 4 replicates in all treatments. So we can have 8 diets, which is a lot of diets, a lot of treatments to be done in one trial. The whole system is about 20,000 liters, and 25% of this volume is just the filtration system, and I'll show a picture soon. And the palatability trials are very fast, about 10 days only, we can have results. We look at SGR, feed intake, FCR, and survival. And we use in this system here for tilapia, we're using 50 animals per tank, which is about 83 fish per cubic meter, which is similar to the density used in cages in river in Thailand. Okay. And same thing, this is the aquarium system for shrimp. We use mostly for shrimp. And in the future, we might use also for fry fish. Okay, about 5,000 liters recirculated. Same thing, 25% of the volume is a filter. And same stocking density, 25 shrimp per aquarium, which is about 83 shrimp per square meter in this case here. So also palatability trial, about 14 days, very quick, and we can do nutrition trial also, same things we look into here. So talking about sustainability, why can we say that this is sustainable? I'm not only talking about sustainability as environmental sustainability, okay? We use very little water, but this is not the main concern. We're talking about social sustainability and also economical sustainability here. So first of all, let's talk a little bit about the construction itself. After all my visits to many R&D facilities, I saw that every time you walk into an R&D facility, you start sweating. Everything is closed, everything is super hot. It's not good for the workers, it's not good for the animals. Well, it depends on the species of the animals we're talking about. So being in Thailand, we know it's always hot. So the idea was, let's make something that's not too hot inside. So the temperature outside in the air will be the same temperature inside here. So if it's cold outside, it's going to be cold inside. If it's hot outside, it's going to be hot inside, but it's never outside the range that the animals can tolerate, okay, for the species we produce here. So if you look at the top, first thing here you have to notice, there's a double roof. So this roof is raised, so there's airflow through that. It's open on the sides, and so the hot air can go out, so you don't feel that heat from the roof. So it's very important. This is a social sustainability for the workers. We want them to feel well when they're working here and happy. Outside also, if you can see, the whole side of the building is not closed. It's all metal mesh. Okay, so animals cannot go in, people cannot go in, but the air can flow through. So there's no heat that gets stuck inside, so it's not very hot inside. Another thing, all the lights we use is LED. So LED light doesn't produce much heat, which is important. And the second thing, it doesn't use much electricity. So economically here is very important. And the second thing, this year we're installing solar panels just to keep the system running during the day. Okay, we're not going to have battery banks or any power bank because of all the heavy metals that comes with the batteries. So we don't believe in that. And also every 3 years you have to change all the batteries. Very expensive. So if you talk about environmentally and economically, it's not viable. Okay, it's not sustainable, this system. So we're going to put a solar panel just to use the electricity when we have sun. If there's no sun, there's no problem. We just use regular electricity. And the third thing is the filtration system. So we're talking about recirculating systems. It's very interesting. So what we have here, we have a drum filter. This is the drum filter. And then we have the biofilter. And all of this is in a big tub, in a big concrete sump here. So it's about 5,000 liters, if I'm not wrong, a little bit over that. No, it's 5,000 liters about. So the water comes from the system through this pipe here. It goes into this drum. What happened with this drum is the drum is closed and the water has to go through this outside. What you see, it's a mesh, a 60 micron mesh. So the water has to go through the mesh and then it goes out the back and into the biofilter. Okay, in the biofilter, there's aeration also to keep all the biomedia moving and all the bacteria alive also. So how this works is, this saves a lot of water and a lot of work because as the mesh, so remember this is half into the water, okay? The drum is just half full of water. As the water levels are going up, this is a water level sensor. It activates, if the water level goes up, it activates this spray bar, which will spray high pressure water against the screen here. And this will rotate this way and clean the inside of the mesh here, the filter. So all the solids and will come out through this pipe here. And from this pipe, it goes into the left side here, which is this canal outside. This canal outside, we have a lot of plants. And here we have the aquaponic system. So we pump the water into the aquaponic system and keep on recirculating here and absorbing all the nutrients. And here's what we get from the aquaponic system. So this is, we were here from the other picture, we were on the back here. The other picture was from this end here. This is a growing bed for like, it's just a hydroponic growing bed. You can have 500 lettuce here, heads of lettuce or any other vegetable you want, but it's about 500. What you see here on the right side is the nursery tanks, okay, the quarantine area. So the water from the tanks also coming here and keeping recirculating here. And this is, those are our technicians there. On the left side, we have a Kun nut harvesting the vegetables. And then on the middle here, Kun Bao also harvesting some of the lettuce and showing the size of the lettuce that we can produce. And as you can see, we use no nutrients other than nutrients that comes with the solids from the production of the fish. So feces and feed, uneaten feed, all ends up here. And there's a lot of fish also inside this canal to make sure there's no bacteria, no insect growing, and also consuming this organic material. Okay. So here on the left side, on the lower side, this is the species that we have these guppies. A lot of these guppies here in the canal, and also in the pond. This is the oxidation pond. Okay, so here we have also guppy. And also we have red tilapia and Asian sea bass to control the tilapia and the guppies also. So it's sort of like a self-sustaining system. There is some aquatic plants that keep on absorbing the nutrients. So this system is closed and ends up here. This is the end of the whole system.
Fabio, can you summarize? It's time with end this.
Yes, actually I'm done. So conclusions. So we know that I did some calculations, but we know that having a commercial production with this kind of system is not yet economically feasible. We cannot compete with people producing tilapia, for example, in cages in a river. Okay, they have much lower cost than this, but it still is a good tool for pilot scale production and R&D. And this should be applied for, I think, for all facilities, because you can sell the lettuce, you can save money on electricity and everything. because we're looking at design that should be maximized for sustainability. So we're talking about economical, social, environmental as usual. And so these add-ons, like I said, the extra savings, and also this financial resource that will come into the facilities come from what you're producing at the facility. Okay, so thank you for your time, for your attention. This is my email if you have any questions, and feel free to ask me any question also. Thank you.
You mentioned about the aquaponic. Can you suggest the kind of if combine with the shrimp culture in marine shrimp?
So in our system, we are using for marine fish with shrimps, we're using white shrimp, so Vaname, and we use 5 parts per thousand salinity. Okay, so very low salinity, and for this kind of production here, for lettuce, we haven't had any problem. I know that broccoli can be used also, tomatoes. What else? I was doing some research, but we haven't had any problem with salinity. Also because there's a big volume of water, and there's also other systems with fresh water that flush into the same system, so it dilutes that salinity. And so we haven't had any problem, but there are options for other species of plants that can be used.
Good afternoon, ladies and gentlemen. My name is Nonapat Jongkaijak. I'm a master degree student from Prince of Songkla University. Today, I would like to present on the topic optimization of stocking density of hybrid catfish in giant freshwater pond polyculture. This presentation consists of introduction, objective, methodology, results and discussion, and the last one is conclusion. As you know, giant freshwater pond is an economically important freshwater species and popular to consume due to their good flavor and firm texture. However, the pond farming still has some fixed limitations such as sexual characteristics, structure, size, early maturation in female, and low production. Therefore, the production and income from pond culturing cannot control. With this factor limitation, polyculture, which the practice of culturing more than one species of aquatic organism in the same pond to utilize all available inch by raising a combination of species having different food habits, feeding behavior, and ecological requirements, and can also increase production and income per unit area. Which type of aquatic animal that were brought up in this research is hybrid catfish. That is the offspring of a male Mekong giant catfish and a female of striped catfish. This hybrid catfish had white-pink flesh color, good in growth and disease resistant, and it can be reared in earthen pond and cage. And the catfish had a feeding behavior in the middle to top level. Therefore, it is possible to culture the hybrid catfish with giant freshwater pond that had a bottom feeder in the same pond. For the aim of this study was to investigate the optimum stocking density of hybrid catfish supplementation into giant freshwater pond in cement tanks. A completely randomized design was used to allocate fortificate treatment into 1 square meter of 12 cement tanks as follows. Treatment 1, 10 pounds per square meter. Treatment 2, 10 pounds with 2 fish per square meter. Treatment 3, 10 pounds with 4 fish square meter. And treatment 4, 10 pounds with 6 fish per square meter. Catfish and prawn were raised in plastic tanks and add the oxygen by aerator. 25 protein pellets was used throughout the, prawn and fish were fed with commercial pellet feed twice a day for 1 week before experimentation. fresh water was added into the tank at 40 centimeter depth, 50 by 50 by 25 cubic centimeter cage will hang into each tank for catfish. A 35 protein sinking pellet for prawn and 25 protein floating pellets was used throughout the experiment. Fish and prawn were fed twice daily. Water quality was controlled by 50% water exchange once a week. Water temperature, dissolved oxygen, and pH were measured once a week. By using YSI Pro 2030 and pH meter. For ammonia and nitrite, measured by using test kit. The sample of each species from each pond were collected monthly, count, weighed, and immediately returned to the water of the same pond. After 92 days of culture, growth measured and calculated according to Burrell in 2002 as follows: percent weight gain, SGR, ADG, FCR, and survival rate. One-way analysis followed by Duncan multiple length test was used to test the effect of stocking density on growth performance. The result of growth performance of giant freshwater pond in monoculture and polyculture with hybrid catfish at different stocking density for 92 days showed that the range of final weights of pond were 13 to 15% of weight gain were 100 to 130%. However, there were not significantly different between treatment in final weight gain, SGR, and ADG. But for FCR of giant freshwater pond with supplemented to fish per square meter were around 3.8, and it was significantly lower than prawn with supplemented with 0, 4, and 6 fish per square meter. In addition, the result of survival rate of giant freshwater pond with supplemented to fish per square meter was significantly higher than other treatment. There were 50%. However, we found high FCR in all treatment of our study. According to the study of New in 2002 mentioned that the growth of small male of giant freshwater pond is stunted by the presence of blue crown male. Feed conversion efficiency seems to be the major mechanism controlling this growth suspension in runt. And the study of Levy in 2017 also found that the all-female culture showed a better performance and feed conversion ratio than mixed culture, and the all-female population exhibit significantly higher size uniformity. So they suggest that for giant freshwater pond, female monosex culture is a sustainable method to yield a homogeneous crop. The result of growth performance of hybrid catfish in polyculture at different stocking density with 10 prawn per square meter for 92 days show that the final weight, SGR, ADG, and FCR not significantly different among the group. However, the result of growth and survival rate of fish and prawn are independent, and prawn were influenced only by their own stocking. Their own stocking range, which correlate positively with yield and negatively with individual growth. In addition, because we have controlled the water quality by 50% of water changing, therefore all water quality parameters were within the suitable range for giant freshwater pond and catfish. So from the this study, we can conclude that the optimum stocking density of hybrid catfish culture supplemented in giant freshwater pond culture is 2 fish per square meter. And it is possible to culture the hybrid catfish with giant freshwater pond in the same pond for increasing production and farmer incomes. Thank you for your attention.
How much giant freshwater pond? How much? How much number of?
Giant freshwater pond.
10
20 to 40 per square meter
per square meter.
for monoculture.
This ratio is still suitable for adult.
I think it's still suitable because in normal, when they mention that the stocking density, they mean that they mean is mean that suitable from fingering until adult.
Good afternoon, everybody. My name is Sunipa Jankel. Today, I want to present topic isolation and characterization of heterotrophic nitrifying bacteria, Acarigines picalis, and efficiency on trim wastewater treatment. Ammonia and nitrite removal in trim aquaculture system are significant to prevent trim from their toxicity. Therefore, nitrification process is the biological process to involve transformation of ammonia to nitrite and further nitrite to nitrate that performed by nitrification. Formed by nitrifying bacteria. There are some bacteria such as Acarigines picalis show a characteristic of heterotrophic nitrifying bacteria in ammonium transformation process. In this study aim to isolate the heterotrophic nitrifying bacteria which have potential to treat wastewater in trim pond. This is a step of the method. The firstly is isolation of nitrifying bacteria. Secondly, 16S RNA gene sequencing and phylogenetic analysis. Thirdly, ammonium removal efficiency in high ammonia media. Finally, ammonium removal efficiency in synthesis trim wastewater. 1 gram of sediment from shrimp farm was inoculated into 100 milliliters of modified pep beef AOM media and checked on a rotary shaker at 170 RPM, 28 degrees Celsius for 28 days. The nitrogen oxidizing activity was tested every 3 days by grease Elotway method. 5 to 7 drops of nitrite reagent were dropped into 1 milliliter of suspension media and less for 1 minute. If the suspension changed to red color, indicate that positive for nitrogen oxidation. Then the positive suspension was diluted and spread on modified pep beef AOM media agar. Culture stain on heterotrophic nitrifying bacteria was obtained and repeat sticking on isolation media. The 16S RNA gene sequence and phylogenetic analysis study. The genomic DNA of heterotrophic nitrifying bacteria was extracted by using the genomic DNA mini kit. The 16S RNA gene was amplified by PCR using the universal primer. The DNA sequence was compared with auto-relay sequence by using the BLAST program within the GenBank EMBL DDBJ database. Phylogenetic tree was constructed by using MegaX program. The ammonium removal efficiency in high ammonia media was tested in flash scale. 1% volume by volume of cell suspension of bacteria was inoculated into 150 milliliters of high ammonium media. In this study, ammonium sulfate was added to 4 grams per liter. After that, checked at 170 RPM, 28 degrees Celsius for 5 days. In the part of mixed strain, we study for 5 ratio. After 5 days for cultivation, the suspension was centrifuged at 3,500 RPM for 40 minutes. To remove bacterial cell, the supernatant was collected and then the concentration of ammonium nitrate were measuring following by the standard method. Ammonia concentration of wastewater was increased by fermenting with shrimp feed for 3 days. After 3 days, wastewater was sterilized by autoclave 121 degrees Celsius for 15 minutes. Cell suspension 1% volume by volume was inoculated into 4 lead to preparing wastewater and aeration and was given throughout the trial period for 14 days. Every day 200 of wastewater were collected and evaluated the amount of ammonium nitrate and nitrate. The result of this study, after resticking on isolation media, heterotrophic natifying bacteria stain SRNB23 and SINB35 were obtained. The colony color of modified peptide on modified peptide AOM media of SRNB23 and SINB35 were creamy and orange. Both of stains were gram-negative and lot-shaped. The identification result based on 16 sRNA gene sequence analysis that SRNB23 and SRNB35 were alkaligenous fecalin, with the sequence similarity value of 98 and 91% respectively. After 5 days of high ammonium media testing, the result provides an evidence of ammonium removal ability of single strain of SRNB23 and SINB35 have ammonium removal efficiency for 91.75 and 91.21% respectively. The nitrite production was 0.09 and 0.04 respectively. The highest ammonium removal efficiency of ratio of mixed strain between SRNB23 and SRNB35 show that at the ratio 30-70 had the highest ammonium removal efficiency of 66.77%, nitrite production of 0.18 milligram nitrogen per liter. However, in this study cannot detect nitrate. The ammonia removal efficiency in synthesis, shrimp wastewater, the results show that the mixed strain of SRNB23 and SRNB35 had the highest ammonium removal efficiency for 63.07%, following by the single strain of SRNB35 and SRNB23. The efficiency of nitrite production in synthesis shrimp wastewater show that all of the treatment were increase of nitrite concentration, except the treatment of mixed strain show decrease of nitrite concentration from 0.15 to 0.4 milligram nitrogen per liter. The single strain of SRNB 35 had the highest nitrite removal efficiency from 0.14 to 0.09. The efficiency for nitrate concentration showed that all of the treatment were increased from day zero except the mixed strain was a little decreased 3.14% from day zero. Discussion of this study. The ammonium sulfate helped to select the heterotrophic nitrifying bacteria because this is toxic to other undesirable species. The phylogenetic tree show that both of two strains are located in branch of alkaligenous ficalate. However, SRNB 35 show a yellow color which different from creamy colony from SRNB 23. Moreover, phylogenetic branch of SRNB 35 show more separately distant from alkaligenase than SRNB 23. Therefore, SRNB 35 may propose to another new species of genus alkaligenase. The alkaligenase ficalate SRNB 23 and SRNB 35 show high efficiency to remove ammonia in high ammonium environment, while autotrophic nitrifying bacteria had limited efficiency in high ammonium environment. Our results are according to other previous studies suggest that alkaligenase provide maximum ammonium removal for 95% with high ammonium concentration above 400 milligram nitrogen per liter. Other reports indicate that alkaligenase ficalate number 4 had a high efficiency for ammonium removal in high ammonium level 2. In mixed culture of alkaligenase strain SRNB 23 and SRNB 35 with a ratio 30 and 70 show highest efficiency to remove ammonia of 63.07%. In this study, lead to report of Joe and co-worker which mentioned that a mixed culture of alkaligenase ficalate number 4 and L1 had ammonium removal rate higher than a single strain culture. Conclusion of this study. The nitrifying bacteria isolate SRNB 23 was identified as alkaligenase ficalate, and SRNB 35 may propose a new species of genus alkaligenase. The mixed culture of SRNB 23 and SRNB 35 at ratio 30-70 show a highest ammonium removal efficiency in synthesis, trim wastewater. Mixed culture of SRNB 23 and SRNB 35 could be suggested to use for wastewater treating in shrimp farm. Thank you for attention.
There are something different between each species.
For the morphology of this species, the morphology, the first thing, the morphology of the two species is different. The SRNB 23 is a creamy color, and SRNB 35 show a yellow color. This is the first one is a different. And in the part of phylogenetic tree show the branch that maybe the SRNB 35 maybe is a different species from the Acarigines ficalid more than SRNB 23.